Good afternoon, everyone, and welcome to the first virtual talk of the weekend. And this is from Randy Oliver, who is in California, where it is earlier in the day than it is here. I'm very pleased to have finally tracked him down, because I've been trying to get him to come and speak to us for some time. And Randy is very well known around the world as a scientist who does practical beekeeping research funded by beekeepers, and then disseminates the results from his experiment and the results from other people's published work around the world. So without any further ado, I'd like to introduce Randy Oliver.
Thank you, Norman. Alrighty, so what I'm going to cover today is Norman asked me to cover a number of topics. So I've crammed about 3 hours of presentations into one and given a very abbreviated versions of them. So we'll see if we have time for more questions later on. So I'm going to focus first on breeding for mite resistance, then on colony nutrition and pollen subs, and then on developing novel sustainable varroa management methods and treatments. So starting off with breeding for mite resistance. And many people are engaged in trying to breed for varroa-resistant bees. And there's various ways of doing it. And most of them are using selecting for specific traits of the bees. What I'm trying to do in California is a traditional breeding experiment. Very simple, not using any kind of molecular biology or single drone inseminations or anything like that, not even serious tracking of bloodlines, just traditional breeding of selecting the stock that has the traits that I want to see each time. So what we do is we go out we start all of our colonies with nukes every spring. And then we let the varroa mites build up in those colonies for a while. And typically by mid-June, early July, mite levels are starting to climb in those colonies. We've given them all one oxalic acid dribble when we make the nukes in the early spring. And we do mite washes on every single colony, about 1,500 hives during the months of June and July, to identify colonies that do not need mite treatments, that have mite counts still of 0 or 1. And any that are that way, we put a tag on top, we do not treat them, and then we will give a treatment to all the other hives, but we don't move any of these hives, they stay in the same yard. We track them for a year, and then the next year we replace all of our queens with queens bred from those colonies that were able to control mites for an entire year without treatment by themselves. So here's how we do it. Each spring, we start about 1,500 colonies from nucleus colonies. This will be all the hives that we will be managing that year. We actually sell another 1,000 nukes on top of that. And we start the varroa race, and we let the mites start to build up. And then that June, July, we do mite washes on every one of those colonies. And if we see 2 or more mites, we eliminate that colony as a potential breeder and treat it. So typically using my varroa model in our colonies here, if we start them in late March or early April, their varroa levels are going to get up into the 50 count, typically middle August into September right here. Our resistant colonies, we want to see them being very, very low counts early on and staying at 0s or 1s through the season until we will allow a little bit higher counts in November. This is something we learned after the first 2 years talking to Dr. John Kiefer. Said, hey, don't give up on them if they spike when all the any remaining mites come out of the brood when they go broodless. And we found that's very important. I just had a breeder yesterday I washed, and she had gone 0s all the way along and then jumped up to a count of 12 mites here and then took it back down to 0 again by the time we're grafting right now. And this is what we're looking for. We're looking for colonies that have 4 to 5 mite counts of 0 over the course of an entire year. This one starting in July. I don't show the last one. We have a number of breeders out, since we're producing queens right now, that have 5 0s in a row for an entire year, right in the middle of all the other colonies with all the mite drift going on. And the thing that we're seeing is that they are beautiful colonies. Colonies without mites are more productive and healthier than colonies that do have mite issues. They're very gentle. We work in shorts and t-shirt with our bees all summer long. Don't need to wear protective gear most of the time. And productive. They're often the most productive colonies in the yard. Notice when their first brood pattern in the spring is usually totally solid. But by later in the season, so this is taken late in the season, we often see this little spottiness right here. And that's apparently due to two traits that they do exhibit. Now, we don't select for these traits, but we notice they do practice uncapping behavior and also varroa sensitive hygiene. It's two different things. So uncapping behavior is when they simply chew off the capping and expose the pupa and the mites to air coming in right here. And Stephen Martin, who is in the audience, he's the expert in this. He has traveled the world, and he can discuss at length this uncapping behavior. But we definitely see this in our colonies. We also see some varroa sensitive hygiene, where instead of recapping these pupae back after the foundress mites exit, that they chew that pupa out completely. And we're making very slow progress with this extreme selective pressure. So we breed off of about 30 queens out of those 1,500 every year. So the other 1,470 their bloodlines are eliminated. So very strong selective pressure, breeding off of at least 30 a year to maintain enough genetic diversity in our stock. And we've gone from a fraction of a single percent of colonies exhibiting resistance, like 2 colonies out of 1,500 to start with. We're slowly working our way up, but it's a very slow progress. We don't claim mite resistance till we get to 95%. So we don't claim we have resistant bees, but every year it is increasing. Okay, next, moving on to colony nutrition and testing pollen subs. In California, we're very much dependent upon pollen subs. Oh, here's first just a warning to you guys that I've seen that beekeepers throughout the world are just suckers for snake oil remedies, for magic in a bottle that they can pour in and all their troubles go away. In our operation, we don't use any of these products, and I've tested a number of them and not found benefit to them. So my suggestion to you guys is if somebody is hawking some kind of health product or remedy to put into your beehives, ask them, could I see the supportive evidence? Can I see the hard data of independent testing that this product showed benefit? And you will find that most of them will be unable to show you hard data. What they tend to do is paid for testimonials. Here's the one that I clipped out of the web somewhere, where the manufacturer of this snake oil product said, they send out emails to all the beekeepers and say, hey, if you have a good testimonial, we're going to give you prizes or free product or something. So there's a competition to see who can write the best testimonial. That is, a testimonial is not the same as a controlled trial. So a question from a number of beekeepers in the US were these probiotic direct fed microbials. So these are probiotics fed in feed directly to the livestock, in this case, the honeybee. Do they produce a measurable benefit to the colony? And the manufacturers make all kinds of claims, great sales pitches. But when I ask them at the conventions for data, they see me coming, they smile, they shake their head. No, we don't have hard data. And I question it because when I look at the actual bacteria included in these different products, none of these come from the bee gut. None of these are the core bacteria found in the honeybee. So anyway, I took 2 of these products to test out, the 2 main products sold in the United States, and fed them per manufacturer's instructions once a month, 2 colonies from July all the way to November. We tested them in one yard that was a sunny yard and one yard that was a shady yard in another area. So we replicated this trial in 2 different yards under slightly different conditions and fed them these probiotics every month. We weighed them and graded them all for strength prior to starting, and then did the same thing afterwards. Here's my grandson who was out of school during COVID and was a very good data recorder and a quality control officer there. I built a custom scale here that we could very quickly get weights to the 10th of a pound. And then we fed them every month with the probiotics, sprinkling them over the top bars. Then what I did is we recruited both the strengths and the honey production, with the blue column being the starting level and the red column being the ending level. So you can see there was not much growth in strengths in these colonies. Now, I was completely blind as to treatment. So I didn't even know which one's probiotic and which one are the controls here, but there was no difference in either between the groups statistically between either strength or honey production. I'm not going to show you all the data, but we did it for each yard and no benefit. I then treated all the colonies with one of two different antibiotics. I know you guys don't use those over in the UK, but I was able to wipe the flora out of the guts with those and then continue feeding them the probiotics. And we then tracked them. I sent the samples off to USDA lab, and they did metagenomic sequencing to see whether the feeding the probiotics helped with the recovery of the gut microbiome, and also if it affected any of the pathogens. And I just got an email yesterday that they're finally going to get a peer-reviewed paper published on that. But the bottom line was there was no apparent benefit either to the gut microbiome or to pathogen resistance. So as far as those two probiotics, I did not find supportive evidence. Now, I'm not saying that if somebody had a probiotic that actually used one of the bacteria that actually is a core bacterium that lives in honeybee guts, that might not be a benefit. But as far as taking all these other animal bacteria and putting them in, I did not find benefit. The next thing is in California, especially the last 20 years, when we've had pretty severe drought, we're unable to keep our colonies alive without feeding pollen subs to them, protein supplements. There's just not enough forage anymore. The summers are just too long and too dry. So in order for us to make our living as almond pollinators, half our income comes from taking strong colonies to the almonds, and we get paid by how strong they are. We feed a lot of pollen sub. So I'm going to give you a very abbreviated version of our pollen sub research. So typical, very dry in the summer, lots of colonies around. I tested out the main subs that are on the market right now. So there's one called the bulk soft, which comes not in patty form, but just in 50-pound boxes that we cut up with a shovel in the yard and feed. We like this a lot because it squeezes between the frames and puts the pollen sub closer to the nurse bee, so it's consumed better. Then Global has a company that makes patty that this one had 15% natural pollen in it. Then Dadant made one called Artificial Pollen 23, AP23. That was fairly new. Mega Bee is a product that developed by Dr. Gordon Wardell from USDA many years ago. Ultra Bee is Man Lake's flagship product, which we've had good luck with before. And then I had a group of beekeepers down in the Southern California area who make up their own homebrew formulation, and they were curious how it would compare in a controlled trial to the formulated products on the market. So they actually were big funders for this project, helped to pay for some of my labor to run this, testing their homebrew formulation. And there's a new one of spirulina algae made, put out by a company called Healthy Bee. Unfortunately, they also added thyme oil from the thyme plant to this, hopefully that it would help with varroa or small hive beetle management. And that actually turned out to be repellent to the bees. So I don't know if it was a fair trial on the spirulina algae, but I'm in contact with the company, and they now sell this without any thyme oil in it, which probably will perform better, but in my trial, it had the thyme oil. Then we did a sham controls. So all of these were about 50% sugar. So we made up a patty of just sugar fondant that was equivalent amount of sugar to here. So we're separating out the benefit of feeding sugar alone from the protein in these patties. We ran 144 hives, 18 hives for each patty type, and we replicated this trial in 3 different yards with different conditions to see if we got consistent results, which we did. And we color coded them all. So we were essentially blinded to treatment. We just went by colors, and we would separate out the, have somebody separate the patties at home and color code them into tubs, the 18 patties, go out to the yard, put the patties, lay them out on top of the hives, have a quality control person at each feeding, go around, make sure they were all properly distributed, and then we would put them in. It's very easy to make mistakes in a big trial like this. So you have to really be careful that you have everything color coded and monitored and quality checked at each feeding. We placed those 3 yards of bees in the worst yards that we, in our history, or historically that we found, where colonies just nosedive during the summer. They're just dry. There's nothing at all for the bees to eat. So we did feed these continually with pollen and syrup throughout the season, all the way from late summer until almond pollination the next spring. And here's the timeline here, starting in mid-August, ending in 6 February, where we expected the dearth to go from here to here, but there was a surprise flow of yellow star thistle. So we stopped feeding. The red means no feeding. The orange means we fed pollen sub. The blue means that we fed sugar syrup. The colonies never lacked for honey or sugar syrup. They gained considerable weight throughout the trial with them. And we graded them at the midpoint, and we graded them at the end point. And when our natural alder flow started here, the first natural pollen coming in, we had to do the endpoint before that first round of brood emerged, which would be 21 days after the alder flow started. So we graded them prior to any influence from natural pollen coming in. We did give them a winter break when they shut down brood rearing, restarted them early in December when we had unusually warm weather, and they started to eat again. So I'm going to skip through all the other stuff, go straight to the end results here. Of those 18 hives of each treatment, I simply graded them all for strength prior to almond pollination and added up the total number of frames of bees, rentable frames of bees, combined for all 18 colonies. And there was a monster difference between the best performing colonies and the sugar controls right here, a number of which just simply dwindled away to nothing or had become very small. So the colonies fed the pollen subs were 3.5 times as, or the best pollen sub, as strong at the end of the trial as the ones that were fed a sugar control alone. So obviously huge benefit to feeding some of these. Now notice that the homebrew and the global way outperformed, are very stood out as best performers. Now I'm going to put a star above those two so you can see. We then had a USDA lab perform analysis of all these pollen subs, look at the gut microbiome of the bee, weigh the head weights and the thorax weights. I'm not going to show you all that data. Again, I'm giving you the abbreviated version here. But notice that the protein content of the subs did not correlate with performance. The 2 that performed best were right in the middle of the protein ranges. The fat content, the vegetable fats and associated sterols that were in the pollen subs also did not correlate with performance. The lowest fat content was on one of the very best performing pollen subs. Down to, generally, we think that you should be up in there, it may be in the 5% range or something. And this is only wet weight down in the 2% range right here. Sugar content, they're all pretty much the same, but again, no correlation with performance. And then here's what really surprised me on the gut microbiomes is that the community structure of the different bacteria that live in the gut was consistent structure-wise all the way across the board, independent about what they were fed. That really surprised me. I assumed that the ones fed with more natural pollen would be since that the gut bacteria evolved with natural pollen rather than artificial diet, that the community structure would be different. But there was no difference in community structure. Also, as far as the total bacterial abundance now, the global patty did have the most abundance right here. Let me make sure I got the right. Yeah. So here, this patty was 15% natural pollen, and this one was 20% natural pollen. But for total bacterial abundance, it was not correlated with the amount of natural pollen. All the rest of these had no pollen at all in the diets. So I'm not seeing that adding natural pollen is necessary for a artificial diet for the bees. And then since we had some colonies that were potential breeder queens in this trial, they did not get treated. They did not get treated with oxalic acid. That's all the other ones did. So I was curious to see if there was any difference in the gut microbiome. And no, continual treatment with oxalic acid in the extended release form did not appear to affect the gut microbiome. That was a good news for us who are working with the extended release oxalic acid. So here is the sad state of our understanding honeybee nutritional requirements, especially the essential amino acid requirements. All that we know pretty much is based upon a study done in 1953 by Antonius de Groot out of the Netherlands. And he did incredible research. I have no criticism whatsoever with his paper. It's an 80-page paper. He did fantastic research, but he was very clear. This was at the very beginning of understanding about essential amino acids and using them in animal nutrition. Every other livestock industry has progressed way, way, way beyond this point and has specific diets with different amino acid ratios for each stage of animal growth. We are still stuck at 1953, and where we're stuck is at the interpretation of this one graph, figure 15. Everything that, all your recommendations that you get about essential amino acids for honeybee requirements comes from this graph, looking at where the inflection point is on these curves when adding a greater percentage of one of these essential amino acids will increase the amount of growth of the bees. And then when it flattens out, that he used this figure. So this would be a figure of 4%. This would be a figure of 2% right here. And a mistake that de Groot made is that because it just so happened that tryptophan had the inflection point at about 1%, he said, well, let's just normalize everything to tryptophan. So all of your amino acid ratios that you see published all are normalized to tryptophan. And that was maybe his only mistake that he made, because tryptophan was not that important as an amino acid, essential amino acid, as is leucine, which is your main amino acid. So what I found is if I normalize all of our diets to leucine, it was far more informative. And you can read about it in my series of articles. I'm giving you the very short version here. So what I did then is I took the analyses of each of these patties and normalized the leucine, relative leucine, looked at what the total amino acid deficiencies were, and came up with this, just a number for your proportional amino acid deficiencies. And when you plot out the performance of the ending total frames of bees per treatment versus the relative amino acid deficiencies, you get this incredibly high R² value, a probability of 2 out of 1,000, 0.0002 of this happening by chance. So very, very strong correlation, indicating that the performance was not based upon how much pollen was in them or any of those other factors. It was based upon how close they were to the ideal essential amino acid ratio. And they tracked right along here. So based upon this, I created an online calculator. That's a free download for anybody. You can get it right off my website. And you can make up your own pollen sub right here by choosing your ingredients or adding them and punching in your proportions. So this would be a 1 to 10 proportion right here. And then it immediately calculates out in the green how closely your pollen sub formation matches the essential amino acid ratios. And what you can do then is you can add type in and say, well, I'll add 1/10 of a pound of lysine concentrate. So you can buy them off the shelf, the concentrates, and bring these green bars up and balance it right out. So anybody anywhere in the world using whatever protein sources are available to you, can now make a perfectly balanced amino acid balanced diet, figure out the formula very quickly using this free software right here. So if you are wanting to feed protein, go ahead and do this. Okay, so let's move on now to the main thing I focused on last year, which is improving some of the varroa treatments to more sustainability. Most beekeepers throughout the world now are using amitraz in some form, and that's because the first two synthetic miticides, the fluvalinate and the coumaphos, the mites very rapidly developed resistance to them. It took about 6 years with the fluvalinate before we started seeing resistant mites, 3 years or less for the coumaphos. Amazingly, we've gotten by on over 20 years or more on this amitraz, and there's apparently a very strong fitness cost to the mites in order to be able to develop resistance. Otherwise, they would have outcompeted the resistant mites by now. But in many areas of the world, amitraz is now failing. So I'm getting a lot of interest from especially large-scale beekeepers who are saying, Randy, help us out here. What can we use rather than amitraz? So a couple of them, and they do that because in our commercial operation, we haven't used a synthetic miticide in 21 years. We run a commercial operation, have healthy bees, we control varroa, but without the use of synthetic miticides. We use the natural miticides, such as thymol. Now, one of the thymol formulations developed over there in UK, I believe, is Apiguard gel, where they take a polyacrylate gel and mix. You don't dissolve. Thymol does not dissolve to any extent in water, but they mix ground up thymol in this gel with the aqueous or water formulation. You put 50 grams on the hive. It works best for us in the hot weather on top of the hive in an 1.5 inch tall rim, and then repeat it as soon as the bees have eaten this up. They have to physically remove this and drag it down through the brood nest for it to be efficacious. If they don't, and I've used fluorescent tracer to show the bees do take this and they do take it right down, not to the sides of the hive, but they drop it right down the middle. And as it gets moved down through the brood nest, the vapors then affect the mites in the brood nest. If the bees do not come up here and physically remove this, it's not due to vaporization up here. It requires physical removal to go down, or it's not efficacious. It is a little bit rough on the colony, especially if you put it between the brood chambers. You will have quite a bit of larval and pupal kill in order to obtain good efficacy, and the bees move their brood nest away from them. So I looked at the different thymol treatments that are sold throughout the world and looked at the dose per application, and they averaged about 12 grams. Apilai-var a little bit less, Thymol-var over in Canada a little higher, and they all require repeated treatments. So Apiguard generally 2 treatments, Apilai-var 3 treatments, and this one has to be repeated also. So they're all problematic in that they require multiple treatments. So what I had is an idea. Well, what if we just put them in a thicker cellulose block? And one of these blocks is a fiberboard wall insulation treatment called Homosoap, which is made from recycled newspaper. And it's fibrous and 1/2 inch thick. So I thought, wow, what if I just take that thymol and dissolve it in alcohol, let it saturate these blocks, and then let the alcohol flash off. In an hour or two, all the alcohol just flashes off, leaving crystallized thymol in this cellulose matrix, this thick matrix. And we tested it out, wondering how it would affect the colony. And we started off with a very low dose, a total of 12 grams per colony, as we saw up above. And boy, the bees didn't get bothered at all. The brood didn't get bothered at all. Did okay mite control, but because they didn't get bothered, I tried higher and higher doses. We tried 24 grams per colony, and then that didn't bother the bees. We tried 36 grams. We finally went up to 48 grams. That'd be the equivalent of 4 individual doses, all applied at once. And even in hot weather, the colonies tolerated, if it was applied above the brood nest, the 48 grams very amazingly well, and it completely zeroed out the mites. We went and inspected the colonies all during the treatments to see how much they moved the brood or whether they stopped brood rearing. We were surprised how easy this was on the bees compared to the other thymol formulations on the market. And it only takes one application rather than 2 to 3 applications. And we did all of our testing in hot weather. We were up there, 35 to 40 degrees centigrade while we tested these. And then I did a very small dose response curve based upon relatively low numbers of colonies. And this would be the amount of percent mite reduction by the mite wash count. And you can see the 12-gram treatment was okay. By 24-gram treatment, looking pretty good. You're averaging 90% efficacy. 36 grams, now you're averaging about 95% efficacy. And 48 grams, 100%, just mites just gone. So probably the sweet spot is somewhere right around maybe 40 grams total. So this season, we're going to be continuing with this. This is not an approved application method. So, okay, Randy Oliver does not recommend or endorse off-label use of any miticides. This is research only. So I will be publishing the findings of what we get out this summer, but I'm very excited about this as a new application method for thymol. And I hope that it gets brought to market. The one thing we're really looking at, though, is the appropriate treatments to use at any time of the year. So there's pros and cons for any treatment. And depending upon what the colony is doing at the time, there's appropriate treatments to use at different times. And typically where we live, where we have almost year-round brood rearing, it takes about 4 treatments a year for us to manage varroa. The tough one is that midsummer treatment when we have honey supers on. The only things that are approved for application then are formic acid, HopGuard, and oxalic. HopGuard does work, but it's very labor intensive and kind of pricey. It takes 2 to 3 treatments to really be efficacious, which is tough to put on when you have honey supers on. Formic acid, if you repeat the dribbles, it's very harmful to the colony. It does suppress colony growth. And if you use vaporization, the approved amount of 1 gram per brood chamber is inadequate for efficacy. It takes about 7 to 10 treatments when the honey super's on to bring the mite levels down well. So, and plus the applicator hazard with the formic. So really, we're very interested in the oxalic acid. We have extensive experience with oxalic acid. This is my grandson, and all of us Californians grow up chewing on what we call sour grass, which is Buttercup oxalis. This is the plant that oxalic acid is named for. And when you're sucking on it, it tastes like lemonade. And it's a mixture of oxalic acid and sugar and water in the sap. So vaporization does work when the honey super's on, but it takes a lot of applications. Dribble works better than vaporization in our studies, but it takes multiple applications, and that sets the colonies back. And I published much more on all this stuff. You can get it on my website. The exciting thing is this idea out of Argentina, and this is the extended release oxalic acid by dissolving it in glycerin as a humectant, and very, very promising. I've been working on this quite a bit for a number of years. And what we found is using the Swedish cellulose sponges, putting in 2½ sponges with our starting mite count being in the blue, even at 48 days post-treatment, you have huge reduction. That'd be the red columns right here. By 70 days, most of these red columns would disappear. You get very, very good mite control. That's a total of 50 grams in the matrix, but that's not the actual dose that goes onto the bees. We'll look at that. The rest of my presentation is going to be on extended release oxalic acid. The beauty is, it does not appear to have any adverse effect on the brood. You really have to put a very big dose on before you see any kind of brood damage. But the 50-gram dose, much of it remains in the sponge. So it's not really a dose to the bees. We'll talk about dose to the bees soon. And I get feedback from beekeepers all over the world sending me photos from their hives and their honey production and their brood of just how successful this treatment is in their operation. This is like going back to before varroa, and really, really does a nice job during the honey flow. I'm really pushing for somebody to get this registered, but it's really a tough process, slow process doing it. So again, I am not advocating anybody using this until it is registered, but this, I'm just telling you the research. The one thing was surprising to us is we had 4 different yards that were in areas where there were no natural honeybee population. So it is either in high elevations, honeybees cannot winter, or one of them was out in the desert in irrigated cropland where there are no other established honeybee colonies. And in those yards where there was no mite immigration from outside colonies, we got incredible results. Look at the lack of red mite counts. So here's your mites per half cup of bees. And look at these high starting numbers, 50 mite wash counts of 50s going down to zeros. This is after 77 days. So you can get incredible efficacy by using around 70 days if you don't have much mite immigration. And this is actual data of mite immigration coming into hives. I have a series of articles going out. I just sent off the final wrap-up article yesterday to the American Bee Journal. It'll be up on my website in 2 months of my study on mite immigration and mite drifting between hives, where we tagged 6,000 bees in 9 colonies intentionally with high varroa levels, intentional to collapse in the yard. And as they were collapsing, we glued painted metal tags on their backs and then put magnetic traps on the entrances of colonies up to 3 miles away and tracked where those bees drifted to with their mites. That's all being published, either out already or will soon be up on my website too. But this is actual data of, these are semi-weekly counts of how many mites were immigrating into hives over time. So this particular colony had a total of 500 mites immigrating into it. So this appears to suppress the efficacy of the extended release oxalic acid. I'm also not just stuck on Swedish sponges. They're kind of expensive. So I'm testing all different kinds of matrices to see what works well. And I've got 2 new ones I'm going to start testing just this coming week. Now, there are a number of questions that people ask me. How safe is oxalic acid for the bees? How and why does it affect the mites? We really don't have a really good idea, other than that if it gets on their foot pads, the sticky empodia on the mites' feet, it will definitely burn the cells there. What is the optimal dose? And is this per hive or per bee? How long does it have a residual action in the hive and affect the mites? Do the residues build up, especially with the extended release where you have it in there continually? Do they build up in the hive? And what's the differences in efficacy? So I'm going to give you some answers to all these questions right now. I'm watching my clock here to see if I run out of time. The first thing is to understand, you don't have to read all this. Pesticidal activity, how pesticides actually work against the pest. There's many different ways that pesticides can work. I've highlighted in red those which may be actions of oxalic acid. Okay, acute toxicity just kills the mites right away. Chronic toxicity, if they are exposed to it day after day, if the deleterious effects build up. Cumulative toxicity, this is like with fipronil, where it builds up in the body, and when it builds up in the body, it then becomes toxic to them. Residual action is how long it lasts. So some things like the organochlorines, they have residual actions of 25 to 50 years, okay, when you put chloridane out there. That's why they've been removed from the market. The residual action of oxalic acid actually is very short, we'll see. And then tissue damage. And this, certainly this is what happens to the mites' foot pads if they walk over oxalic acid crystals. And then we'll talk about behavioral disruptors later on, which may not be toxic at all, but affect the behavior of the pest. So here's the frustrating thing. We've used oxalic acid for over 30 years for varroa control, but we're completely blind still on how it gets distributed and persists inside the hive. So let's take a look at the dose per bee. If I look at the 3 approved application methods by Apibioxal, the dribble application, the spray application, and the vapor application, if you follow the label, how much total dose, and then divide by the estimated number of bees that's specified on the label, it comes up to be for the dribble about a little over 100 micrograms. A microgram is a millionth of a gram per bee. With the spray application, again, right around 100 micrograms per bee. Vapor, it's 50 micrograms, but we know that is not efficacious. It really takes 100 to 150 micrograms for a vapor application per bee to be efficacious. So the take home here is this number 100. Keep this number in your head for the rest of this presentation. That 100 micrograms per bee is the theoretical dose per bee for applying oxalic acid to control varroa. 100 micrograms per bee. So the questions are, how big a dose actually shows up on the bees, and how long is its residual activity? So let's first look at it with live bees. And here I used a green fluorescent tracer that glows under black light, and an hour after dribbling a colony with oxalic acid, you can see the bright green glows on these bees, on their bodies. Very irregular, it's not smooth at all. It varies tremendously from bee to bee. And then I made a special box just for photographing under black light. So again, this is an hour after application, and I got arrows pointing to the visible green specks right here, where this bee got a lot on its wings right here. This one got on its wings, on its bodies. This one's on its back underneath its wings here. Another glow over here, little tiny glows. This is how it gets distributed very, very unevenly on the bees. The other way to look at it is by acid-base titration, to actually measure how much acidity is on the bees. Now, we're not measuring oxalic acid, we're measuring acidity, but since the oxalate ion has been shown not to be effective against varroa, it's apparently solely the acidity. So, but that's probably valid to simply measure the acidity. I'm converting all these acidity measurements to oxalic acid equivalents, how much oxalic acid it would take to get to that, in micrograms, to get to that equivalent. So I take test tube, you add an indicator that changes color. This one changes from orange to blue at low pHs. And then drop in a bee. If it changes towards orange, that means it had an acid on it. And then what you do is you drop in a calibrated titrant, drop by drop, counting the drops. So I can make a titrant being 1 microgram per drop, or 10 micrograms per drop, 100 micrograms per drop equivalent, until it goes back to the starting color. And now you know by counting how many micrograms of oxalic acid are on that bee. This totally opens our eyes right here, and it's very, very accurate. So here what I did, just as an example, I mixed up an oxalic acid solution that would be 10 micrograms per drop. So this is by weighing the oxalic acid into a liter of water. You know exactly how many micrograms of oxalic acid are. I put the 10 drops, so I knew there were 100 micrograms in this test tube right here. These two outside ones are our starting solution. So you can see the color of the starting solution. Now, I then added a titrant, also calibrated for 10 micrograms per drop. I put in 9 drops here, and you see it turned it from orange close back to here, but not quite. If I put in 10 drops, exactly match. If I put in 11 drops, it overshoots. So this shows how accurate I could very accurately titrate, saying it took 100 micrograms of titrant to neutralize, to bring this back to the starting mic count. So this, we can get down to single microgram determination. Back in 2018, when I first developed this method, I realized, wow, it's not consistent. If I leave these dead bees on the bench for a few days, the acidity disappears on dead bees, okay? No, so it wouldn't be bacterial action, it wouldn't be grooming action. So we this last year we did experiment to see how rapidly oxalic acid degrades once on a dead bee. So we killed bees one morning, dipped them into a oxalic acid solution, dumped them out, let them dry on a paper towel, and then either left them out in the room or put them in the freezer. We also vaporized a hive, and you can see the oxalic acid crystals on the setae, the hairs of the bees right here. And a close-up. This is what oxalic acid looks like on a bee. Here's a bee with a dose of maybe 60, 70 micrograms per its body. That's how much you would see on the bee's hairs there. And then we put the oxalic solution on plastic cover slips. And this is for 0 hours up to 12 hours. This would be a day, 24 hours, then 2 days, 3 days, 4 days. We tracked them for 12 days. Notice there's no change to the oxalic acid when the water evaporates off on the cover slip. But if you add either glycerin or sugar to that water, don't even put it on the bee, it rapidly, very rapidly degrades. It gets neutralized, apparently by reacting with the sugar or the glycerin. If you put it on bees, so here's vaporization, it also largely drops down by the second day, you're down to half the dose or less here. I'm sorry, this is the vaporization with the red. This is on the oxalic acid glycerin on plastic here. But notice how rapidly, within the first 12 hours, you have incredibly fast degradation of the oxalic acid, the acidity, on dead bees with no grooming, no movement, no bacterial action. So this opened our eyes. We realized we needed to, you can't freeze samples and sample later. We scientists are very used to being able to just freeze samples and then analyze them later. You can't do that with this. Putting them in the freezer did not help. So I threw out hundreds of frozen bee samples. I had Ziploc bags from previous research, realized they were no good anymore. So we had to start all over again in 2022. So first thing I did, I went out to a colony that had oxalic acid sponges in it for 73 days, May through July, in hot, dry weather. Curious whether the oxalic acidity built up on the bees. So we set up the titration tubes, all the same color, and placed 3 bees from that hive into each tube. And also checked bees in a hive sitting next door that had not had any oxalic treatment, and there's no color change at all. We've never seen any color change at all with untreated bees. As you can see, based upon the starting color, there was clearly acidity on those bees, all pretty consistent, about 2 to 3 micrograms. Very, very, very low after 73 days of continual treatment. The question is, so obviously it doesn't build up on the bees, but how can such a low dose be efficacious? So you may be curious, how much oxalic acid is a microgram? So I put in a scoop and pulled off this little tiny crystals right here and dropped this into a test tube. And that's the color change from that little tiny crystal right there. And I titrated out 65 micrograms. So that's 65 micrograms of oxalic acid right there. That's very little. And that's about as much as you would see on a well-hit bee after an oxalic acid dribble. Now that we had this dialed in, we set up 15 colonies and apply oxalic acid by a different treatment method to every colony. So we did a vaporization. First did a 3 gram per brood chamber, 3 times the allowed rate. And then we followed it by 1 gram. 3 different kinds of dribble that was repeated, 7 different kinds of sponge treatments, and then several New Zealand strip treatments and a shop towel treatment. And I went back out hour by hour to start with, and day by day, took from the same marked frame from every colony 3 samples of 5 bees to titrate. And you can see the color change. So we're getting averages now per bee for 5 bee samples. We've changed now. I'm doing all single bee 10-day samples, but for this trial, we did 3 5 bee samples. I hired a new assistant. This is Rose. And first we started out right next to the beehives so we could titrate them immediately. And then it got too hot there. So I set her up in a trailer adjacent to those hives, a workstation with a fluorescent light so she could do rapid titration. And there she is titrating a batch of bees right there. So I'd take the samples of the bees, bring them in, and she'd titrate as I did that. We collected a huge amount of data on this. And then finally, graphed it all out. So all our different treatments. And I'm going to go over these. This was our retreatment for the dribbles and vapor. This is the retreatment for the New Zealand strips. The point of this graph is notice most treatments, you get a spike, not up to 100 micrograms, okay, but up there a bit. And then it holds fairly consistent as it goes along for pretty much all of them down to that 2 to 10 microgram level. So we are now unblinded. I can now tell you exactly what happens as far as the acid on your bees. So let's break it down by doing the vaporization. Notice there's a couple of bees that get hit really hard. And I titrated one of these out to over 1,000 micrograms. So that will kill the bee. Most of them are much lower than that. And then also the dribbles too. So here's the vaporization. The red is the 3 gram per brood chamber vaporization, and then followed by a 1.5 gram per brood chamber vaporization. So we got about half the dose. But notice how quickly by day 1, a day later, you've dropped down to almost nothing for the vapor. The dribbles, you start off with actually a higher dose per bees, and they actually stay high simply a little bit longer than the vaporization. The main thing with the dribble, you do get a higher initial dose on the bees. It's not quite as well distributed on the bees, but it is a higher dose. The different sponge applications, okay. And we also tried citric acid, which looked pretty good, but we did not test this for efficacy, only for residues. And all the different applications were fairly similar. It didn't seem to make a whole lot of difference how much acidity on the bees by the application method of them. And then we did this New Zealand strips using different ratios of oxalic acid and glycerin. So either the 1-to-1 ratio of oxalic to glycerin, 1 to 1.5, or 1 to 2.5, which is the Argentinian dilution. And again, not a whole lot of difference between the ratios. You get a spike up there your first few days, and it starts to drop off. And when I plot out just the different ratios, it didn't seem to make much difference. That was surprising to me too. So a lot of surprising answers to these questions when we actually titrate all the bees. Now, one of the issues with vaporization and dribble is that they wear off so fast. You have all these bees still emerging from the brood. And when I do a calculation on a colony with a mite wash count of 25 and half a cup, okay, so that'd be about an 8% infestation. You have about 650 mites emerging from the brood every day. So if you drop off your efficacy of treatment after 2 days, which you do with dribble or vaporization, you still have all these mites emerging from the brood for the next 12 days. So that is the issue there. And you also can see how that tracks out with the extended release treatment, where you get a fairly high mite drop at about 2 days, and then it continues to knock down mites as they emerge from the brood for the next 12 days or so, until it finally gets very low. So we're understanding how that extended release works. When I model it out of putting in 0% mite kill per day, we get a normal buildup of mites. If I put in a 5% kill of mites per day, it pretty much holds the mite level flat. If I'm killing 10% a day, it's looking like after a month, I've dropped down to less than half the mite population. At 25% kill a day, you would actually eliminate most of the mites within a month. Extended release appears to be around this level, somewhere around maybe a 10% kill a day. So how does it work at such low levels? So here I've done individual bee titrations after a dribble of 10 bees. and look at the difference in acidity levels across the board compared to untreated bees where there's no change at all. So very erratic distribution on the bees. And I have much more data published on this. And then I found this paper that helped me a whole lot. Nazi and Milani, researchers out of Italy. I have yet to meet either of them, but I have tremendous respect for their research over the years. He figured out how much oxalic acid was needed per square centimeter of the bee's body. So I could go to a paper that had computerized tomography of the surface area of a bee's body and calculate out that when we're getting an average of 32 micrograms per bee, you would expect to have a kill after 48 hours of close to 100% of the mites. If you're down to that 3 to 4 microgram level, which we see with the extended release, you'd be expected to be killing about 45% of the mites that walked over a bee at that level that were exposed for 1 hour to it, about a 45% kill after 24 hours. So this validates that even that low level on the bees could have this kind of toxicity to the mites. And we're getting short on time, so I'm not going to show you. This is breakdown of individual bees now after dribble and vaporization. Let's skip past that. And one more potential mode of action is a behavioral disruptor. And this is how we control tracheomite with Crisco. The vestible shortening confuses the young mites when they're moving out of one bee and trying to get the other one. They can't identify a young bee by smell if there's Crisco in the hive, and they're unable to successfully reproduce. It doesn't kill them, it just stops them from invading a new bee. And this could be the case with varroa. If you cover up their sensory papillae on their front legs right here, they are unable to sense the right age bee to crawl onto. So I set up a bioassay here where I could dribble some bees with oxalic acid to get a homogenized sample of bees, divide them in two, dribble half the oxalic acid, put them in the incubator overnight to let it react, and then add untreated bees from the same batch to treated bees here. Then collect live mites using powdered sugar and drop a bunch of mites into the middle and see which way they walk. I was only able so far to do one test, and they mostly all walked in one direction. And then it was late in the season, and my last high mite hives died from varroa. So I'm waiting to build up some mites in a couple of hives right now so I can continue with this behavioral assay. This would be a real eye-opener if we find out that one of the modes of action of the oxalic acid and glycerin is that it creates an odorant, an ester, an oxalic ester that confuses the mites. So the only treatments we've used for all these years, my point here is we don't need breakthroughs or high-tech chemicals. We already have these three tools. If we learn how to use them and expand the labels, we can get by quite well with varroa control. The key thing is to rotate treatments. Don't just use one treatment all the time. So in our operation, we rotate formic, thymol, and oxalic. We've done it for many years, and you can maintain the efficacy of the treatments. This would be off my first year beekeeping, shows how we treat our hives. And that is the wrap up there. If you have time, I'd be happy to answer specific questions.
Yep, no, that's great. So the question was about the nucleic acid content of the origin of the protein supplements. So
okay, so I've just added that to my model this last two. Oh, it's not even on the website yet. Okay, if you want to get that, remind me. I'll have to update my website because I just worked this out. So I put in, what you want to do is have 40 to 50% of your protein content being in the essential amino acids. Over that is not a benefit, and straight essential amino acids is harmful to the bees. So you're aiming for the 40 to 50% level of essential amino acids relative to total protein. And my updated model now has that, but I don't think I've posted it to my website yet. I'll have to do that this week. And if you need it right away, I'll be happy to email it to you.
So the question was actually about the nucleic acid content of the different protein sources.
Oh, the nucleic acid.
Yes.
I have no idea. if they have nucleic acid, or if it's even important, nucleic, yeah. I mean, because the honeybee animals make their own nucleic acids. Okay, so when you are eating protein, you digest it down to the essential, or to the amino acids, and then you reformulate your proteins and nucleic acids out of them. So there's not a whole lot of nucleic acids that pass directly through the gut. So I'm not, I can't really answer your question.
So the question was about leucine being a branch chain amino acid, which is widely used in sort of protein shakes for bodybuilding and so on. Have you seen an effect of other branch chain amino acids on bees?
The only thing I've looked at, just the ratios of those amino acids that we know are essential. So whether they're branch chains or not, they're either essential or they're non-essential. So if they're non-essential, the bees make their own. So all they need is just the essential ones. So as far as how they're branched, I don't know that really has a whole lot of relevance to creating a diet.
Okay, we have a question right at the back. So firstly, he was thanking you for your efforts over many years. And then he was saying, when would you expect the approval of the sponges that you were talking about?
Well, the IUN cap is already approved in a number of South American countries. I was recently down speaking in Peru, and it's sold there. They are pursuing some other registrations. I have there's two guys that I know of in the United States, beekeepers, who are pursuing registration, and I've been in communication with the EPA on this, and the EPA moves about the speed of plate tectonics. It's really, really frustrating. You say, well, we got to check the supplier. Well, to check the supplier, it's got to take a minimum 16 months just to even check the source of the oxalic acid before they even start looking at registering it. So extremely frustrating. I just wrote up a white paper for the benefit of the beekeepers and the EPA in the United States of what legal and what legal workarounds and what illegal methods beekeepers can and are using, just to say, hey, here's the bottom line, that the regulators can even work with us, or there's going to be a lot of people just ignoring it. So New Zealand was brilliant. New Zealand, the government of New Zealand understood beekeepers are going to use these things. So they said, tell you what, we're going to pass you an own use exemption. You can use generic oxalic acid, thiamol, formic acid, any way you want. You take full responsibility. Don't worry about it. You can't sue us. Your fault, it's on you. Beautiful. It works beautifully in New Zealand. So in New Zealand, the manufacturers can't sell a formulated product, but they sell you a kit that has glycerin and oxalic acid and the strips, and you just make your own. That's totally legal in New Zealand. And we're going to be pushing for that in the United States, because beekeepers are pesticide registration. They're in the US, they're only concerned if you're going to cover more than 300,000 acres with a pesticide. If you took the total number of bee colonies in the entire United States and put them side by side, it would cover 30,000 acres, just a tiny bit of room. So we're going to try to get some exemptions specifically for pesticide applied to beekeepers, because the purpose of in our country, FIFRA, the Insecticide Act, is that there is no unreasonable risk to man or the environment. Right now, they're well aware that with thiamol, formic, and oxalic acid, there's no unreasonable risk to man, the environment, to the honey contamination. They all have exemptions for tolerance. So my question is, why are they even involved in it? They know there's no unreasonable risk right there. Just let us free with those only for the bee industry, applied within beehives rather than sprayed all over the landscape.
Okay, on that note, I think we ought to let Randy go now, and thank him once again for
all right, thank you.